Okay, good afternoon, everyone. Welcome to lecture 41 of BMS 300. Today we're going to start with the composition of blood, and we will move on to talk about the cells involved in the immune function in tomorrow's lecture. All right, and just so you remember, we have exam 3 tonight, right? And whatever I discuss starting today all the way until your Thanksgiving break starts will all be on exam 4. All right, so with that, let's jump right in. So here's an overview of how our discussion today on the blood and will actually fit into the other organ systems we've been discussing. So the very first unit, we talked about the main component of a cell, and the main component of a cell is water. And so we see that the blood is also mainly made up of water, and also a number of proteins which are found in the blood plasma in addition to the water. And we talked about how proteins are made in unit 1. We saw how central dogma explains how the information in the DNA is moved to the RNA, and from the RNA we have proteins being made. And we also talked about the general structure of a cell in unit 1. And when we talk about blood, we'll see that there are different cells we'll find there, and the major cell we'll be talking about is the red blood cells, or the erythrocytes. And the interesting thing is the erythrocytes are not true cells because they don't have the organelles which you would expect to find in a typical cell, and so we'll see why that is the case. And because the red blood cells don't have mitochondria, the way they make ATP has to be only through anaerobic metabolism. It cannot use the oxygen to make ATP. And in unit 1, we moved on from the structure of a single cell to cell junctions to see how various cells are connected. And there will be no discussion of cell junctions when we talk about blood, because each of the cells, they are found suspended in the fluid matrix, and they're not linked to one another, so there won't be any discussion on cell junctions. But when you talk about different types of tissues, we see that we talked about 4 types of tissues: connective tissue, epithelial tissue, muscular, and nervous tissue. Blood will fall under the category of fluid connective tissue. And from the types of tissues, we moved on to the discussion of the integumentary system, and we will notice that the integumentary system will play a very integrated role in the immune function of the body. Now, for the immune function of the body, a number of organ systems will play a role, but the integumentary system is the first line of defense. And as shown in this little picture here, you probably can't see all the details, but by the time we're done with the immune system, you will know what this little yellow box contains. It shows you the innate and immune response when you have, let's say, a splinter go through your skin. And from the integumentary system, we moved on to the skeletal system, and we find that the skeletal system plays an important role in the immune function, because both the red blood cells and the white blood cells are all made in the red bone marrow, which is found in the bones. And from the skeletal system, we moved on to the endocrine system, and we find that the endocrine system's function is to make hormones which are sent through the blood to various target tissues where they will bind to receptors. So if you have any problems in the blood, that will even have an impact on the functioning of the endocrine system. And from the endocrine system, we moved on to the nervous system, and the main role of the nervous system is to detect changes in the environment and to respond, and even changes inside the internal composition of the blood can also be detected, and appropriate changes can be made. And one example of this is the presence of both peripheral and central blank. What is it? What receptors? Chemoreceptors, right? That's on the exam for tonight, right? So we see that the central and peripheral chemoreceptors can detect the composition of the blood, and they will make appropriate changes. If you have a drop in oxygen levels, or if you have an increase in carbon dioxide levels, specific nerves will be activated, like the phrenic nerve controlling the diaphragm, or the intercostal nerves controlling the intercostal muscles, and it can modulate the rate of breathing accordingly. So we see that even the nervous system will keep an eye on the blood composition. And we see that the muscular system also is going to function mainly based on blood being pumped to it, because the blood will bring oxygen, it will also bring other nutrients, it will carry away waste products. All of that is possible because blood is brought to the muscles. And the way the blood is brought to different parts of the body is through the cardiovascular system. So we see the heart is going to pump the blood through the blood vessels to the various parts of the body. And one of the parts to which the blood is pumped specifically for oxygenation is the lungs. So blood is brought to the lungs where it can be oxygenated, and when blood is taken to the rest of the tissues, it will be deoxygenated. So this brings us to what we have discussed until this point, right? And even moving forward from here, when you try to integrate the various systems, blood is going to be the key, because we'll talk about the digestive system to see how the nutrients enter into the bloodstream. We'll see in the urinary system how the blood will be purified, and finally in the reproductive system we'll consider how there are a number of reproductive hormones which are going to be transported to target tissues and how reproduction happens, right? So this is how blood fits into what we are discussing as a whole in this semester. So here's an overview of what I hope to cover today, right? We'll talk about the various components of blood. So we see that blood composition can be divided into two parts. You have the blood plasma, which is in this supernatant, and formed elements, which is mainly made up of the red blood cells or erythrocytes. And we will be talking about how these red blood cells are made in a process known as erythropoiesis, and we'll also see how once the life cycle has come to an end, how these red blood cells are broken down, and how the various components inside the red blood cells can be recycled for making more red blood cells. And after our discussion of the red blood cells, we'll talk about white blood cells, or also known as leukocytes, and this is a topic which we will have to discuss tomorrow, right? But we see that there are different types of white blood cells: neutrophils, lymphocytes, monocytes, eosinophils, and basophils, right? So each of these white blood cells have some unique functions, and we'll talk about it. And tomorrow I'll tell you what is the connection between all of these cells, these 5 different cells, and this monkey eating a banana, right? So that's something you will have to wait until tomorrow for us to get to. And once we talk about the white blood cells and the red blood cells, we'll discuss the functions of blood, and we'll move on to talk about the immune function of how the blood plays a role in mediating immunity. So the question is, what is blood, right? So blood is the specialized connective tissue that contains cells suspended in a fluid matrix. I told you that any type of connective tissue you consider, there will be cells which are held in a matrix, and in the case of blood you have a fluid matrix where these cells are suspended. So how do we know what is in the blood, right? You can take the blood sample and you centrifuge it, you will have this supernatant, which is called the blood plasma, and the blood plasma is going to make up around 55% of the overall blood composition. But I want you to notice that there is a range at which the blood plasma can exist. It can be as low as 46 or as high as 63, and this really depends on the level of hydration. See that the blood plasma is mainly made up of 92% water, and so if you are overhydrated, you would be in this higher range, but if you are dehydrated, you may be in the lower range, right? So what are the other things in the blood plasma besides water? We also have around 7% plasma proteins, and one example of this is albumin, and I will briefly talk about albumin in the upcoming slides. It can bind to certain substances, but one of the main functions of albumin is to ensure the osmotic pressure in the blood. There are other transport proteins also found in the blood, and that could be helped with the transport of hydrophobic hormones, right? For example, thyroid hormone is hydrophobic. In order for it to be transported in the blood, it has to bind to plasma proteins. And around 1% of the plasma composition would be other solutes. So this could include electrolytes, wastes, metabolic waste, and other dissolved substances in the blood will be forming the 1% of the plasma. So moving down to the formed elements found here in the pellet, we see that the formed elements can have two parts. You have this narrow region called as the Buffy coat, and it's going to contain the white blood cells and also blood platelets. So blood platelets is something we won't be discussing in detail. The one thing I'll tell you is that whole blood clotting is possible because of blood platelets. If you remember back to the integumentary system, we talked about the various steps in the wound healing cascade, and the very first step in the wound healing cascade is hemostasis, and the blood platelets will play a role in blood clotting. And from the white blood cells and the blood platelets, we move on to the major components of the formed elements, and that is the red blood cells or erythrocytes. And this will make up around 99.9% of the formed elements, and the Buffy coat will be less than 0.1% of the formed elements. Now there is a term called which we use to describe the percentage of red blood cells in a given blood sample, and that is called hematocrit, right? And this term hematocrit, the percentage of red blood cells within a sample of whole blood, it's a term used in the clinical setting, and I'll give you an example of how we use that term as far as blood doping is considered, right? So we'll see that later in today's lecture. So now let's look at the structure of a red blood cell. So what you see here is a two-dimensional view of a blood sample, right? This is a light microscope image. So you take the blood sample, you stain it, and you squish it between two slides, and you'll notice that the red blood cells appear flattened here, right? And the one thing which I would draw your attention to is that there is a lighter color in the center of the red blood cell, and that's called a lighter pallor. And the reason for that lighter color in the center of the red blood cell is because it has got a concave structure. There's a small dimple in the middle. If you look at a three-dimensional structure, you see these little depressions or dimples a lot more clearly, and if you see a cross-sectional view, you notice that it has actually a biconcave structure. It's concave on both sides, and so it forms a biconcave disc. So a couple of things about the dimensions of a red blood cell. So the thinnest part of this biconcave disc will be around half a micrometer to 1 micrometer in thickness, whereas the thickest part of this disc will be around 2 to 3 micrometers in thickness, and even the diameter will be around 7 to 8 micrometers, right? So the reason I give you these dimensions is not to have you memorize it, but just appreciate how there is going to be a large surface area to volume ratio. So the given surface area, because of this biconcave shape and also the small size of these cells, you have a large surface area to volume ratio, and that is very important for the oxygen to be quickly moving in and to move out of the red blood cells. There's another thing which is possible because of this biconcave shape, right? And that is referring to the fact that these red blood cells can form stacks. So here's a stack of red blood cells shown right here, and this stack is also known as a rouleau, and the purpose of this stack formation, like stacking of dinner plates, is so that it can pass through narrow blood vessels. So notice that here you have a blood vessel with a diameter which is only slightly larger than the diameter of a given red blood cell, but because they're able to stack through, they can pass through these blood vessels. Now if they did not stack, they would collide with one another, and that can result in a log jam taking place, and so there could be a blood clot forming, and blood can't flow through these narrow vessels. Also interesting to note that the red blood cells, they are quite flexible, and so they can bend and flex when they are in a narrow area, so that blood flow can happen without any problems. So notice that these cells are highly specialized, and one of the specialized features of these red blood cells is that they have no nucleus, and so the term used to describe a cell without nucleus is an anucleate cell. Also, this does not have any mitochondria, and the absence of mitochondria means that the red blood cells cannot use oxygen to make ATP, and so all of the oxygen which is carried by the red blood cells, they are only going to be transported from one place to another. None of it can be used by the red blood cell to make ATP. Also, there are no ribosomes, and the lack of ribosomes means that no more proteins can be made. There's no nucleus, so there's no DNA to make mRNA, and even if there was mRNA, you don't have any ribosomes to make proteins. And as a result, if there's going to be any sort of damage to the red blood cells, it can't be repaired, and the cells can divide. And so it's understandable that the red blood cells have a very short lifespan of around 120 days, and after that they are broken down. So now let's move on to talk about one of the major proteins you will find packed in the red blood cells, and that is hemoglobin. So this is a slide which should be a review for all of you, I hope, because the exam for tonight is on hemoglobin and how it helps to transport oxygen and carbon dioxide. So yes, this is the major function of hemoglobin, to help in oxygen and carbon dioxide transport. Notice that it's got a quaternary structure, meaning it's got multiple subunits which come together to make a single hemoglobin molecule. And so there will be two alpha subunits. They're shown here in purple, both are identical, and there will be two identical beta subunits shown here in blue. And at the center of each of these subunits you will find a heme group, right? So the heme group is shown here, and it's got an iron in the middle of a protoporphyrin ring. So protoporphyrin ring is this complex structure shown here, and the good news is I'll never ask you to memorize it. There may be other classes where you might have to do it, right, depending on your future goals if you're taking a more advanced course. So the iron is in the middle of this heme group, is where oxygen will bind, and when oxygen binds to this iron, it's called oxyhemoglobin, and when oxygen will unbind from the iron, it's called deoxyhemoglobin. So the only way, or the major way in which oxygen is transported, is 98% is by binding to the iron in the center of the heme group. Around 2% will dissolve directly in the blood plasma. Carbon dioxide, on the other hand, will be carried by a different mechanism. It won't bind to heme, but it can bind to these other alpha beta subunits instead. And I hope you're all familiar with the 5 factors which can affect oxygen binding to hemoglobin. You could have changes in carbon dioxide levels, pH, temperature, the fetal isoform, and 2,3-bis-phosphoglycerate. All that can affect oxygen binding to hemoglobin, and we talked about it ad nauseum in part 3 and 4 of the respiratory system, right? So my hope is most of you have already learned it, right? And if you haven't learned it, I will test you on the same concept in exam 4, right? Because blood is a part of the hemoglobin structure is considered even in this unit. So we'll test you on exam 4 on the same material, and we will also be tested on that in the final, right? So if you've learned it once, more bang for your buck. Okay, now let's talk about erythropoiesis, right? What is the various steps involved in the synthesis of the red blood cells or erythrocytes? So I want to draw your attention to the red bone marrow. Red bone marrow is found in the spongy bone, and in the red bone marrow there is a region known as an erythroblastic island, right? So what you're seeing here is an erythroblastic island, and in the erythroblastic island you have these hematopoietic stem cells, which is abbreviated as HSC. And these hematopoietic stem cells, they are going to differentiate into a proerythrocyte. Okay, so the question is, what causes the hematopoietic stem cell to differentiate into a proerythrocyte? And it is due to interleukin 3, which is released by macrophages in the erythroblastic island, right? So notice that this interleukin 3 is a type of cytokine which is just acting as a messenger for cellular signaling, and we'll talk about other types of cytokines later in today's lecture, right? So we will come back to what cytokines are later in today's lecture. So once this proerythrocyte is made, it needs something else to move on to the next type of cell in the formation of red blood cells. And there are these cells known as pericytes, which are found in the kidney, right? So this pericyte is the cell which has got this number of projections arising from it. These cells are sensitive to the level of oxygen in the blood. So keep in mind that the kidneys will filter the blood, so as blood flows through the kidneys, these pericytes can detect if there is a drop in oxygen levels, and if so, it would produce a hormone known as erythropoietin. So erythropoietin is also abbreviated as EPO. And EPO will bind to a receptor on the proerythrocyte, and that will cause the proerythrocyte to further differentiate. All right. What are the various steps involved in this differentiation? I've indicated it by 3 arrows so that you don't have to know those details. Well, just keep in mind that there will be changes happening in the proerythrocyte until it forms a normoblast. Now, a couple of things to notice in this normoblast is that it's got a receptor for erythropoietin, so erythropoietin will bind to the normoblast as well. Secondly, notice that there is no nucleus in the normoblast because it has kicked out its nucleus. So there's no nucleus in the normoblast, and it also gets rid of the other organelles as well. And this normoblast will turn into a reticulocyte. And in this reticulocyte, it's going to contain around 80% of the hemoglobin which would be found in a mature red blood cell. So the question is, why is it called reticulocyte? And if you observe a reticulocyte under the microscope, you'll see these network-like mesh structures found inside, and that is basically the mRNA which is being used to make hemoglobin, right? So that is the reason why it's called a reticulocyte. And the reticulocyte will enter into the blood capillaries in the bone marrow, and from there it will mature into a erythrocyte. And the erythrocyte, for this maturation to take place, will take a couple of days, and the changes which take place include a remodeling of the cell membrane, and there will also be a volume reduction where it's going to take this biconcave shape, and any remaining organelles in the reticulocyte will be exocytosed. So some of these remaining organelles will be exocytosed. So a couple of days after a reticulocyte enters into the circulation, you really can't differentiate that from a mature red blood cell because these changes have taken place to the reticulocyte. Okay, so these are the various steps involved in erythropoiesis. Right. Are there any questions at all before we proceed in this slide? Okay, there are no questions. We need to consider what is blood doping and how EPO can play a role in that as well. So blood doping is a dangerous practice used by some athletes to reinfuse red blood cells to elevate the hematocrit. So hematocrit, as I mentioned previously, is the percentage of red blood cells in a sample of whole blood. And there are some athletes who could take their own blood and freeze it, and during the race maybe reinfuse it back into their bodies, and that can increase the hematocrit. Others could also use what is known as erythropoietin. Now the original reason why erythropoietin was synthetically made in a lab setting using recombinant techniques was so that those who have kidney failures, the blood could be purified using dialysis, right? But if they don't have these pericytes to make erythropoietin, then they will not have the red blood cells being generated, and that can lead to very low energy levels. And so that's the initial reason why erythropoietin was synthetically made, to help people with kidney failure. But then athletes used this, and they found that it can help increase the performance, right? And the way it increases performance is a twofold way. Firstly, it's going to increase parameters like oxygen consumption. So if you have more red blood cells, you will have more oxygen being transported, right? More red blood cells means more hemoglobin to bind to the oxygen, and so you'll have a increase in the oxygen consumption. And if you have more oxygen being used, that means you can make more ATP, and if the body is making more ATP, it's going to take a longer time for exhaustion to occur, right? So more ATP means it's going to increase the time for exhaustion, and that is the rationale behind which athletes were using EPO to increase the hematocrit. But many of these athletes were caught doing it because the type of EPO they used was made in a cell line known as the Chinese hamster ovary cell line, and those cells will have a different type of will ensure that there's a different type of post-translational modification on the EPO, right, which is different than the EPO found in humans, and so that's how they could catch these athletes and ban them, right? But I just want to point out that this is called as a dangerous practice because it can lead to a number of consequences. Firstly, it can thicken the blood, because if you have a greater hematocrit, the blood viscosity is going to go up, and that can place a tremendous stress on the heart to pump this viscous blood throughout the body. I already mentioned that the resistance for flow is going to be influenced by viscosity, and viscosity can be increased by increasing the formed elements in the blood. Notice there also a risk for autoimmune diseases, because when you use synthetic erythropoietin, which is made in the lab, you're going to introduce foreign proteins into the body, and that could trigger an autoimmune response. And I said that there could be heart disease because the heart is under greater stress. There can also be a stroke or a pulmonary and cerebral embolism, and embolism is just referring to the blockage in the flow of blood because of an air bubble or even a blood clot. And you could have these blood clots developing not only in the lungs, and if it happens in the lungs, the lung tissue will die. It can even block blood flow to the brain, and that can damage the brain tissue as well, right? So this is a dangerous technique, and athletes have found a way around this by just reinfusing blood into their own blood, right? So people can't detect that something foreign has entered in. And so, do you know what is the way to track people who are doing that? So modern times they have what is known as, recently they have this athlete's passport, biological passport, where they will keep track of a number of parameters in the blood, like the normal hematocrit levels, what is the level of testosterone in their urine, and so they will keep track of this over time, and if there will be any abnormalities and changes in the concentration of any of these parameters, right, they will see that potentially they may have been engaged in illegal practices. So that's how the sports authorities try to keep athletes in check. But the question is, does EPO really help in endurance, in doing better in endurance activities, right? So I want to show you this study which was done a few years ago, and yeah, let's see what they found.
EPO is a well-known doping drug in many sports, including cycling. It stimulates the production of red blood cells, which carry oxygen to the muscles. Therefore, cyclists think this could improve their performance. To investigate this, the Center of Human Drug Research studied the effects of EPO on cycling performance in well-trained cyclists. In total, we included 48 cyclists, of which half received EPO injections and the other half placebo injections. On April 4, 2016, participants started the study that would last 11 weeks. They received one injection of EPO or placebo every week for 8 weeks, and in this period performed 2 types of laboratory tests to measure performance. A maximal exercise test leading to exhaustion was performed before the first injection and every 2 weeks during the treatment period. For the other test, participants performed a time trial of 45 minutes, a sub-maximal exercise test. This test was done twice, once before the first injection and after 7 weeks of treatment. So, let's see how the cyclists did. Baseline levels of VO2 max during the maximal exercise test were similar for both groups, but already after 2 weeks, a difference started to show in favour of the EPO group, which persisted over the entire treatment period. A second maximal performance parameter, maximal power output, showed a comparable effect of EPO. In the sub-maximal exercise test, mimicking a time trial, both groups started out at the same level as well. In contrast to the maximal test, however, after 7 weeks of treatment, there was no difference observed between the 2 groups in this test. Although these 2 laboratory tests measure several aspects of cycling performance, real races are won on the road, not in the laboratory. So, to verify the effect of EPO on road racing, our participants raced the Mont Ventoux at the end of the treatment period. After a 110-kilometer course, each participant raced to the finish line at the top of the climb. The average time of both groups was calculated, and in this real-life road race, the EPO group did not perform any better than the placebo group. So, in one overview, although we saw an effect of EPO on maximal exercise performance in well-trained cyclists, this effect was much less clear in a sub-maximal time trial-like test and completely disappeared in a real-life road race to the top of Mont Ventoux.
Okay, so the bottom line is that in a laboratory setting, there was going to be an increase in high-intensity outcomes, right? For example, like the amount of maximal force generated, or even the amount of oxygen which is consumed, doesn't seem to be higher when they did a high-intensity activity. But when they looked at an endurance activity, which is where you will expect the EPO to have an impact to increase the overall oxygen available to make ATP, there wasn't very big difference. And in the real world cycle, real life cycle race, they didn't find a difference between those who used EPO and those who did not. And so the question is, did those athletes who used EPO, did they actually have a benefit by using EPO and increasing their hematocrit levels? Right? The question is still out there. Anyway, but now let's consider how the red blood cells are broken down. So as the blood flows through an organ called the spleen has 2 regions. It has a region known as the red pulp, and then there's an area known as the white pulp. The white pulp is where you will have immune cells, and we'll talk about its function when we come back tomorrow. But in the red pulp, you will have macrophages, right? They are these immune cells, and they are going to detect the level of oxidation on the glycoproteins which are found on the surface of red blood cells. So if the glycoproteins on the surface of the red blood cells have a greater level of oxidation, that means they've seen enough life, they're probably worn out, and so those cells are going to be phagocytosed. If the level of oxidation on these glycoproteins isn't as much, then the red blood cells won't be phagocytosed. So once phagocytosis takes place, the question is, what happens to all of the components which are found inside the red blood cells? So what happens to hemoglobin? So for hemoglobin to be recycled, there are a number of organs which play a role, and one of it is the bone marrow, where you have red blood cell formation. You will also have the macrophages in the spleen which play a role. Notice even the digestive system gets involved, because there's the liver playing a role and the large intestines are involved, and the urinary system finally is also involved in excreting some of the byproducts from the breakdown of hemoglobin. Okay, so let's see how this happens. So notice that the red blood cells, once they are mature, they are released into the bloodstream, and after around 120 days or so, we see that a small amount of the red blood cells can just hemolyze, or they can rupture in the bloodstream. So the 10% of these cells, the components such as the alpha and the beta heme chains, they will be broken down in the kidneys, and the heme will be excreted in the urine. So they are sent to the kidneys, and from there they are excreted in the urine. But the majority of the red blood cells, around 90% of that, will go to the spleen, where they are phagocytosed, and all of the amino acids which are released from the breakdown of those alpha and beta chains, they will be sent back to the bone marrow for making more hemoglobin. But the heme subunit in the middle of those chains, they will be broken apart to have the iron taken back to the bone marrow by a specific protein that's called transferrin. The name isn't necessary, but notice that the iron is taken back to the bone marrow. But the protoporphyrin ring, that will be converted into biliverdin. So biliverdin is this pigmented compound, and as the name indicates, it's got a green color to it, and biliverdin will in turn be converted into bilirubin in the macrophages, and bilirubin has got this orange yellow color, and bilirubin will be transported to the liver by the protein called albumin. I mentioned albumin briefly when I talked about the proteins in the plasma. So albumin not only plays a role in maintaining the osmotic pressure in the blood, but also can help in some transport as well. So bilirubin is taken to the liver, and from the liver we see that it is going to enter into the bile, and that will give bile its characteristic color as well. We'll talk about the function of bile when we discuss the digestive system and fat metabolism, but eventually the bile can enter into the large intestine, and in the large intestine you have a number of gut bacteria which will further break down bilirubin into either urobilins or stercobilins. It really depends on the type of bacteria. Now urobilins has a yellow color, whereas stercobilin is brown in color, and depending on the percentage of urobilin versus stercobilin, the color of the feces will differ, right? So it really depends on that proportion. Now some of the urobilins can also be reabsorbed back into the bloodstream, and since the blood is filtered in the kidneys, it can enter the bloodstream, it can enter the urine, and it will give urine its characteristic yellow color, right, depending on the level of hydration of that individual. Now there could be problems in the liver which is going to affect the excretion of bilirubin, and that can lead to a condition known as jaundice, where you have yellow coloration of the skin and even the whites of the eyes, right? So we talked about jaundice briefly when we were discussing the integumentary system, but this is what jaundice would look like in an advanced case. So now we have completed all I have to discuss about the red blood cells, and we will consider a few definitions before we talk about the white blood cells and how immune function is carried out. So the first definition I want to introduce you to is this term called PAMPs, which stands for pattern associated molecular patterns, right? So this is just referring to the molecular patterns which are found in nature, and they will signal to the immune system the presence of pathogens. And an example of a PAMP would be lipopolysaccharides, which are found on the surface of gram-negative bacteria. So depending on the proteins which are found on various kinds of pathogens, they can serve as PAMPs, and those patterns can be recognized by the immune system. Now, the second definition is DAMPs, which refers to damage associated molecular patterns, and this damage is indicated by a high level of certain compounds which should not be found in the extracellular space. An example of this is the presence of ATP. So ATP levels are not high outside the cell, but if you have cells which die, they will rupture and dump their ATP out into the extracellular fluid, and that can be detected by the immune system that something is going wrong. We will see specific examples of how PAMPs and DAMPs play a role in mediating the immune response. Another definition is CD. The term CD you will see a lot, and it's referring to the cluster of differentiation. And these are just proteins found on the surface of immune cells, and they have a twofold function. The first function is to help identifying a given cell. For example, if it has CD4 protein on it, you say it's a CD4 cell. If it has CD8 on it, you'll say it's a CD8 cell. And secondly, it can also help in cell-to-cell signaling as well, right? So it helps in cell signaling and in the identification. So here's the quick example of how there can be cell identification and cell signaling as well. So notice here in this first panel we have an infected cell shown here in orange, and then you have an immune cell shown here in green, and this is a T cell. And it is also known as a CD8 cell because it is expressing the CD8 protein on its surface, right, shown here in pink. Now the CD8 protein is what allows these two cells to come in proximity, right, because the CD8 will bind to the MHC1 protein shown here in green, and that allows these two cells to interact, and this immune cell is going to release perforins which is found inside it, and these perforins will pump holes into this infected cell, and as a result the infected cell will die, right? So I hope you can appreciate how there is cell signaling, and also there is you have the identification of a cell based on the type of CD protein expressed on it. We'll come back into the details of how this actually happens when we talk about innate and adaptive immune response. So the last definition I want to bring your attention to is the term cytokines. So cytokines are proteins which will function as signaling molecules, right? And we see there are three types of signaling which can take place. You have autocrine signaling, where a cell is basically talking to itself. It releases a certain molecule which binds to the surface of the cell, which releases it. Or there could be paracrine signaling, where the released molecule will bind to a cell in the close by vicinity, or endocrine signaling, where the released signaling molecule is going to be sent through the bloodstream to a target organ. So cytokines can have different types of communication they mediate. So examples of this include growth factors, right? We see that when growth factors go to a specific type of cell, they may differentiate. We will look at an example of this tomorrow. We also talked about interleukin 3, how that will be released by macrophages, but that will signal to the hematopoietic stem cell to differentiate. Can also lead to chemotaxis, when chemokines are released, right? And I'll explain what I mean by this. So when you have cell motility to a region where there's a damage, that is what is chemotaxis. And we'll see how various immune cells are going to come to the region where there's a damage, right, to trigger the defense response from the body. And I said there can also be differentiation which takes place, and the example is when the hematopoietic stem cell will differentiate into a proerythrocyte in response to interleukin 3. And there will also be alteration in gene expression. So I want to explain how all of this happens by reminding you of a slide which you saw a long time ago. Do you remember when we talked about this slide? Was it unit 1 or 2? Can be 3 because that's on today's exam, right? Unit 1, yes. And where did we discuss it under unit 1? Was it under cell junctions? Yes. What type of cell junction? Tight, gap, anchoring. It was under tight junctions. So we talked about how when there's, so what you're seeing here is an villi in the small intestines. And when you have dysfunction of claudins, you can have the tight junctions not functioning properly, and as a result, pathogens can get in between these cells. And so when pathogens come in, notice that there are these immune cells, macrophages and dendritic cells, which we'll talk about. They produce cytokines, and here is a list of the cytokines they could produce. It's a number of interleukins, and these cytokines will lead to chemotaxis, where you have various T cells arriving here, and those T cells can cause more cytokines to be released, like other interleukins, and these will in turn cause inflammation, and then as a result it can cause the gene expression to be affected. And in this case we see how claudin expression is affected. Alright, so I highlight this slide to you just to show two things: how cytokines work, and also to tell you how integrated the course material is. Right, you really can't learn it and forget it. Right, it all comes back and it integrates. So with that, we will conclude today's lecture. We'll come back and talk about the white blood cells tomorrow, and I wish you all the best for tonight's exam. Right, and I'll be available here to answer any questions you may have.